evening, my respected sir, ma'am, and good evening to all. This is Anindita speaking from PG 4 Durgapur Government College. First, index of the project. First, introduction, objectives of the project, methodologies used, main findings, conclusion, and reference. Second, introduction. Since this project is about bioactivity compound, so we need to know what is bioactivity. ম্যাম শোনা যাচ্ছে? What is bioactivity? Bioactivity refers to some biologic activity such as biochemical or immunologic reactivity or the biologic effects of the substance. Next, my chosen compounds are lysine, second as oxaloacitic acid, and third riboflavin. First, lysine. Lysine is an amino acid essential for animals and human nutrition. And lysine is produced today only by microbial processes, and a variety of approaches for its production have been developed. And this is the lysine structure of lysine, and UPAC name 2S26-diaminohexanoic acid. Second, oxaloacitic acid. Oxaloacitic acid is an intermediate of the citric acid cycle, where it reacts with acetylcholine A to form citrate, catalyzed by citrate synthesis, and it is also involved in glucogenesis, the urea cycle, the glycoxylate cycle, amino acid synthesis, and fatty acid synthesis. And this is the structure of oxaloacitic acid, and UPAC name 2-oxobutanoic acid. Third, riboflavin. This is also known as vitamin B2. Riboflavin is a type of vitamin B complex and which is essential for metabolic energy production. Riboflavin is a carbohydrates, fats, protein into glucose for energy, and UPAC name 4,8-dimethyl-10 2S3S4R 2345 tetrahydroxypentyl benzoate di2,4-diene. Biological function of riboflavin. Riboflavin works with the other B vitamins. It is important for body growth. It helps in red blood cell production. Riboflavin is essential to formation of two major coenzymes, flavins, mononucleotides, short form FMN, and flavin adenine dinucleotide, short form FAD. FMN is synthesized after the addition of phosphate in riboflavin, and FAD formed by the transfer of an AMP moiety from ATP to FFN. Next, objectives of the project. And we have used ChemDraw and Password software to analyze the bioactivity of the chosen compounds, and we have done the literature survey of these compounds and explained them in this project. We found the following useful bioactivities of this compound. First, lysine. Lysine main findings: first, fibrinolytic protein kinase inhibitor, and third, antioxidant. Second, oxaloacitic acid. Its main findings are alanine transferase inhibitor, oxygen scavenger, and third, catalase inhibitor. Next, riboflavin, vitamin B2. And main finding: leukopoiesis inhibitor, insulin secretaceous lysozyme inhibitor. Methodologies used. First pass online website, then registered to way to draft and predict the chosen compounds and 6 bioactivities prediction of the chosen compounds. Main finding: lysine. Useful bioactivities of lysine. First, there is fibrinolytic. Fibrinolytic is a normal body process. It prevents blood clots that occurs naturally from going and causes problem. Fibrinolysis is a highly regulated enzymatic processes that prevent unnecessary accumulation of intravascular fibers and enables to the removal of thrombi. Next, protein kinase inhibitor of lysine. A substance that blocks the action of enzymes called protein kinase. There are many different types of protein kinase and they take growth and division. Uses. Blocking certain protein kinase may help keep cancer cell from growing. Some protein kinase inhibitors such as imatinib, vermouthilib and gabapentin are used to treat cancer. Next, antioxidant. A substance that protects cells from damage caused by free radicals. Antioxidants are molecules that fight free radicals in our body. Free radicals are compounds that can cause harm if their levels become too high in our body. Use. They are linked to multiple lines including diabetes, heart disease and cancer. Our body has its own antioxidants to keep free radicals in check. Next, oxaloacitic acid. And main findings are alanine transferase. First, an alanine transferase inhibitor. Short form ALT. ALT is a transferase enzyme. It is also called alanine aminotransferase and was formerly called serum glutamate pyruvate transferase and was first characterized in the mid-1950s by Arthur Karman and colleagues. ALT is found in plasma and in various body tissue, but it's most common in the liver. It catalyzes the two parts of the analysis cycle. Serum ALT level, serum AST levels in their ratio are routinely measured clinically as biomarkers for liver health. This is the structure of alanine transferase inhibitor. Next, oxygen scavenger. An oxygen scavenger is the substance that scavenges oxygen chemically and therefore protects the packaged food completely against deterioration and quality changes due to oxygen. A mixture of iron power of calcium hydroxide scavengers both O2 and CO2. Removal of O2 can be used fully cured poultry meat products since multiple hardly then exist to prevent outgrowth of C. botulinum spores. Next, catalase inhibitor. Catalase is a peroxisome specific marker proteins belonging to the catalase family. It is the very important enzyme in the protecting cells from oxidative damage by ROS. Drugs bind to enzymes and leakage enzyme inhibition. One such example, catalase inhibitor by lead to H2O2 accumulations and cytotoxicity in the cancer cells to H2O2 mediate NF-kB suppressors and apoptosis activation. Next, riboflavin, vitamin B2. Main finding, leukopoiesis inhibitor. Leukopoiesis is a form of the hematopoiesis in which the white blood cells WBC are formed in bone marrow located in bones in adults and hematopoietic organs in the fetus. White blood cell. Indeed, all blood cells are formed from the differentiation potential. These immediate cells, lines or colonies are progenitors or red blood cells, erythrocytes, platelets, megakaryocytes, and the two main group of WBCs, myelocytes and lymphocytes. Next, insulin secretaries. Insulin secretaries are the diabetes medications indicated for the treatment of type 2 diabetes. The role of the riboflavin in the glutathione redox cycle is considered as is the indirect oxidative action. Adam et al. Have reported that riboflavin reduces fasting blood glucose level in a dose-dependent manner while it increases calcium and GLUT4 expression as calcium is required for insulin secretion. And last, lysozyme inhibitor. Lysozyme inhibitor effectively protect bacteria against lysozymes challenges and thus could contribute to host colonizations or infections. Further, more they can be antiseptic to modulate the host immune response by interfering with the release of PG fragments during infection. And my conclusion, there are many opportunities that lysine, oxaloacitic acid and riboflavin present on the market